I hope I'm visible and audible now. Earlier it was not. So today in this video, I will be discussing about the unit of chapter 5 NCRT. Yes, fine. So I will be discussing about the chapter 5, that is an principle of inheritance and variation. Alright, so this is a very important chapter for your NEET examinations. You can expect at least 5 to 6 questions from this entire chapter. So it is under the unit of genetics, and let me start by introducing myself. I'm Janani Saptharishi and I'm currently pursuing my BTech Biotechnology, and I'm associated with Unacademy. I will be teaching class 12 biology, and yes, this is what you will gain with Unacademy. You will have daily live classes, all right? You can wish to crack any entrance exam you want to, and we'll make sure that we will prepare it to you. And we have the best features that are these daily live classes with your educators, and it is a very interactive sessions. We'll engage in doubt clearing sessions, question answer polls, and there are many features to it. We also conduct live tests and quizzes. All right. So we have a very structured courses. If you join with my referral code, it doesn't mean that you will get access to only the contents of biology. No, you, if suppose you are preparing for an examination that is NEET, that means you can avail all the content of physics, chemistry, biology, and all the necessary subjects required to crack an examinations. So we have a very structured courses that is in line with your syllabus, and we promise you, you take any time and duration course, your entire syllabus will be completed. And also we have unlimited access, that is one subscription will get you access to all the live and recorded courses to watch from your all at your comfort, your ease. All right, so you will have these live classes and you will be on board with the top educators of India. So, in such a pandemic situation, this is the best scenario we can imagine, just sitting at home and having access to all the content, all the information, and you can really make the most out of it. We have a very comprehensive syllabus. All you have to do is download, install the Unacademy Learning app, and get a subscription. And once you're done with that, for more information, please type this URL in the Telegram app and you can click on join. All this information will be provided in the description box below. So as soon as you'll get subscription, these are the courses that are offered for you. So this is the fee and this is the duration. So I recommend you the most the 12-month one. All right, but because I will be teaching your biology for preparing you for the NEET examinations. So for an examination like NEET, you will at least need 12 months in hand to get going. And if you're in your class 12, I will highly recommend you the 6-month or the 3-month one. All right. And not only this, the 12-month package is the most economical. You can get live test, quizzes, take mock test, and you'll get unlimited access to all the videos. And you will, if the charge is only rupees 15,000 for 12 months. And for an additional gift from my side, it is that if you use my code, that is Janani, my name, Janani, all right, use this as your referral code and you can avail an extra 10% off on all the courses and the fees. Fine. So let us start. In this video, I will be discussing the chromosomal basis of inheritance. That is how the Mendel described many laws. We saw that it had some loopholes, not exactly loopholes, some deviations from it. Like the law of dominance has something as incomplete dominance. And so now we will see that how two scientists proposed that the chromosome is actually the basis of inheritance, and also how it resembled very much with that of the laws of Mendel. But though it resembled, why was the chromosomal basis was accepted readily, but not the Mendel's law was not accepted very readily. Now it is very recognized, but in those times it was not. And also, we will learn in this video about a unique concept called as Cleotropy and how it will affect some genes, and we will learn two diseases relating to it, very important, that is sickle cell anemia and phenylketonuria. So if you have any doubt, please put it up in the chat box and I'll make sure to clear all your doubts. Hello FZN. So we often see that all the children attain most of their characters from their parents, because our mother and we the genes flow from one generation to the other. That is why children resemble their parents in various characters. The example of the characters can be eye color, skin color, body shape, etc. So the transmission of characters from one generation to the another, this process is called as heredity. All right, एक जनरेशन से मतलब एक पीढ़ी से दूसरे पीढ़ी में आपके कैरेक्टर्स फ्लो हो रहे हैं. तो ये एक एग्जाम्पल है डिफरेंस इन कैरेक्टर का. तो आप देख सकते हैं कि हम सभी like humans में we have different skin colors. It is even regarding to like geographical conditions also, and even very much physical and genetic conditions also. So we have a varied variety of everything, and there are even differences in characters between parents and their offsprings. Now this is called as variation. All right, the parents are sorry, the offsprings are not exactly similar to the parent. All right, they will have some variation, but some of the characters will resemble that to the parents. So let us start by learning how the scientific study started and how this mechanism of hereditary and ये variation कैसे cause हो रहा है. ठीक है, ये सब जो भी हम पढ़ते हैं ये सब include होता है genetics में. And the father of the genetics is Sir Gregor Mendel. All right, we saw in my previous video how he carried out experiments in the pea plant. All right, and how he proved that it followed a particular fashion, that is the law of dominance. We saw the law of segregation and law of independent assortment. So this is a very good diagram of the DNA, that is as the genetic material. All right, and this basically shows that how the DNA is packed here. And yes, so this is basically all the base pairs of the DNA. This is the double helix structure. Then it is coiled into histone proteins. It becomes the nucleosomes is formed, and then we will see that, yes, see, and then finally the chromosome is formed. So this chromosome is basically highly condensed form of the DNA. And where is it present? The chromosome is present in the nucleus of the cell. All right, so this is its location. Very important, please don't get confused. Chromosome is present in the nucleus of the cell. uh, I've seen many people writing chromosome is present in the DNA. No, it is a highly coiled form of DNA where present in the nucleus of the cell. So, as I said, highly condensed or coiled forms of DNA. So, the method by Mendel to distinguish between heterozygous dominant and the heterozygous recessive is known as a reciprocal cross. We had discussed this in last video. This is just a question based on that. So let us start with our main topic. So these Walter Sutton and Theodore Boveri, these two scientists have a major contribution, or they are the one who have developed the theory of chromosomal inheritance. Hi Arun Patil, right? So ये जो chromosomal theory of inheritance है, इसको वॉल्टर सुटोन एंड थियोडर बोवेरी ने प्रपोज किया था, ठीक है। तो इन्होंने कहा था कि क्रोमोसोम है जो एज अ कैरियर एक्ट करता है एक प्रोसेस ऑफ हेरिडेटरी में, या फिर फॉर द ट्रांसफर ऑफ जेनेटिक मटेरियल फ्रॉम वन जनरेशन टू द अनदर। और हमारा जेनेटिक मटेरियल क्या है? तो आवर जेनेटिक मटेरियल इज द डीएनए, ऑलराइट। So, the nucleus of the gametes contains the chromosomes, and which will carry all the hereditary characters. Alright? And it is a link between the parents and the offsprings. So, there can be a questions framed that what is the link between the parents and offspring? So, what is that link? That link is the chromosome. Hi Bharat, how are you? Fine. So, as I said, that it is basically a link between two generations. Now, coming to how chromosomes are, where are chromosomes found? So, chromosomes are found in pairs in somatic cells, and in it is also found in sex cells, all right. But what happens is in somatic cells there is a recombination, all right. So, this is how the recombination means first there is combination, and again it is rearranged, all right. So it undergoes first meiotic division, and you can see here it has fused, or I would say, into one another. And again there are four haploid formed, and they undergo yes, so this is how the homologous chromosome pair, and then they segregate or separate independently at meiosis, all right. And morphologically and genetically they are identical. So thus each gamete will contain only one pair of chromosome. I hope this is clear. So what happens that during the process of fertilization, the union of the sperm and the egg, now see, now we have seen that कि वो haploid बन गया। तो अभी ये देखो, तो haploid बन गया, इसका मतलब ये नहीं कि next generation में 23 ही flow होगा 46 में, नहीं। तो फर्टिलाइजेशन के प्रोसेस के वक्त वापस वो अपना रीस्टोर कर लेता है अपना ओरिजिनल नंबर, दैट इज 23 plus 23, that is 46, all right. So they undergo a process of meiosis, and during fertilization they will restore their number. So who supported the chromosomal theory? We saw that pictures of two scientists. Out of that, one was Boveri, Sutton and Boveri, all right. So the chromosomal theory of inheritance, also very commonly called as Sutton-Boveri theory. Remember this name because it will also include the name of the scientist, which can again be a very probable question. So the chromosomal theory of inheritance, that is the Sutton-Boveri theory, what it what it states, that it identifies, that it states that chromosomes are the carriers of genetic material, all right. तो ये कह रहे हैं कि क्रोमोसोम ही एज अ कैरियर एक्ट कर रहा है, जो हमारे सारे जो फैक्टर्स है या जीन्स है या जो भी ट्रेट्स है वो एक जनरेशन से दूसरे जनरेशन में पास हो रहा है। यही सेम चीज मेंडल ने कही थी, बट मेंडल ने क्रोमोसोम टर्म यूज नहीं किया था, एंड मेंडल मेड हिज प्रेडिक्शंस मच बिफोर, ऑलराइट। And at that time there was not even a microscope, so nobody could imagine such a structure. And when Sutton and Boveri produced these terms, there was a microscope was discovered. We have seen how a cell looks. We know what is the nucleus. All right. So it became somewhat acceptable, and we knew what is chromosome. Like it was, we could relate to it, and hence this theory was readily approved. Is theory of Boveri, and this is fundamentally unifying the theory of genetics. So unifying means he basically connected the Mendel's law and proposed this as the chromosomal theory. So basically he unified or combined it. All right. So it states that the chromosomes are the one that are seen in all dividing cells, right? So chromosome nucleus में होता है, and अगर कोई भी cell divide करती है sexual reproduction के केस में, तो वो उसके लिए एक जेनेटिक मटेरियल होना जरूरी है, ठीक है, एंड वो कैसे होता है, क्रोमोसोम से. तो इट इज प्रेजेंटेड ऑल डिवाइडेड सेल, ऑलराइट, and it will pass from one generation to the another. This we will see how, all right, and are the basis of the all the genetic inheritance. Fine. So who did the experiment and who verified that yes, chromosome is passing from one this to another? So this was done by T. Morgan. Uh, we know that he conducted the experiments on Drosophila or the fruit fly. So his name is Thomas Hunt Morgan. All right. So, uh, what he did was he studied this chromosomal basis on a Drosophila or a fruit fly. All right. This experiment discovered the basis of what is known as the variation that a sexual reproduction produces. All right. What is the scientific name of a fruit fly? It is Drosophila. So this is how exactly a fruit fly looks. And why did he choose a fruit fly? Why not any other fly, any other insect? तो सबसे पहले जो मेजर एडवांटेज है फ्रूट फ्लाई का, इट कैन बी ग्रोन ऑन अ सिंपल सिंथेटिक मीडियम इन अ लेबोरेटरी. इसका सिंपल वर्ड्स में ये मतलब है कि इसे हम इजीली लेबोरेटरी कंडीशंस में ग्रो कर सकते हैं. हमें कोई एक्सटर्नल फिजिकल फैक्टर्स की ज़रूरत नहीं पड़ती. ठीक है. और एक एग्जांपल ये भी ले लीजिए, जैसे हम सिंपल सा यीस्ट हम ग्रो करते हैं या कल्चर करते हैं अपने लैब में. सिमिलरली उसी उसी अमाउंट के मेहनत के साथ हम एक फ्रूट फ्लाई को भी कल्चर कर सकते हैं. ठीक है. एंड अनदर इम्पॉर्टेंट एडवांटेज, इट विल कंप्लीट इट लाइफ साइकिल इन 2 वीक्स. तो हमें सिर्फ 2 वीक्स वेट करना है इसके जनरेशन को देखने. So we need not have a larger waiting period. It is reduced, all right, and we can come to a conclusion or we can get results quickly. Fine. सो अ सिंगल मेटिंग, ठीक है, जब भी ये मेट करेगा तो सिंगल मेटिंग में ये प्रोड्यूस करता है लार्ज नंबर ऑफ प्रोजेनीज. ऑल राइट. तो अब मैं आपके इसके क्रोमोसोम्स को समझाती हूँ. जैसे नंबर ऑफ क्रोमोसोम्स इसमें केवल 4 होते हैं. ठीक है. तो उसमें से 3 जो है वो ऑटोसोम्स होते हैं और एक पैर जो है वो सेक्स क्रोमोसोम होता है. ऑल राइट. तो जैसे देखिए ह्यूमंस में कितने 23 पेयर्स है, उसमें से 22 पेयर्स क्या है, ऑटोसोम्स, और सिर्फ एक सेक्स क्रोमोसोम है. So it is very similar here. Alright, so even this have one pair of sex chromosome. Now another major advantage, just like humans, alright, just like humans, even their female have a ploidy of X and X, alright, they have a homologous X chromosomes for the uh, sex chromosomes of female, and the male have X and Y. So this is another advantage for choosing Drosophila. Alright, so its entire scientific name is Drosophila melanogaster. So this was the exact organism that was used. So this is the adult female and this is the adult male. Alright, so see, ये हुआ फर्स्ट पेयर ऑफ क्रोमोसोम, ये हुआ हमारा सेकेंड पेयर, और ये हुआ हमारा फोर्थ पेयर. और इनमें से सेक्स क्रोमोसोम कौन से होते हैं ये हम देखते हैं. तो सिमिलरली ये हमारे ये देखिए जो फर्स्ट पेयर है ना जहाँ पे एक्स एंड एक्स लिखा है, ये है सेक्स क्रोमोसोम फीमेल्स का. ठीक है, अडल्ट फीमेल का. फाइन. एंड मेल का भी हमने यहाँ पे देखा. हाँ, ये हुआ मेल का पहले सेक्स क्रोमोसोम आया, ये था हमारा फर्स्ट पेयर. देन वी हैव द सेकंड पेयर एंड द थर्ड एंड द फोर्थ पेयर. सो दे आर ऑटोसोम्स. तो मॉर्गन के एक्सपेरिमेंट ने क्या कहा कि द एलील्स फॉर द विंग साइज, ठीक है, इन्होंने टेस्ट किया था विंग साइज, दैट इज वो लॉन्ग है कि वेस्टिजियल है यानी कि शॉर्ट है. ठीक है. एंड द एलील्स फॉर द बॉडी कलर, दैट इज ब्लैक और ग्रे. फाइन. Black body vestigial winged fly or the gray body long winged fly. So ये दो characteristics को use किया गया. All right. So what happens in Morgan's experiment is that it yes, so it produces, what happens is that it will produce all long winged gray bodied flies in the F1 generation. All right. Now then they will अभी एक बार हमें F1 जनरेशन मिल गया तो जनरली मेंडल के केस में हम क्या करते थे, हम करते थे इसकी सेल्फिंग, ठीक है। मतलब उसको जैसे पी प्लांट का जो F1 जनरेशन है उसको वापस से सेल्फ पॉलिनेट करते थे उस हेट्रोजाइगस कंडीशन को। बट यहाँ हम क्या करेंगे इज वी विल नॉट डू सेल्फिंग, बट यहाँ पे F1 जनरेशन हमारी जो भी आई उसको हम क्रॉस करेंगे वाइल्ड टाइप के साथ, ठीक है, उसको क्रॉस किया जाएगा वाइल्ड टाइप के बाद, and we let them mate with the wild type, that is the F1 hybrid female with the black vestigial winged male। तो हमें यहाँ पे F1 में क्या आया था, लॉन्ग विंग्ड ग्रे, ठीक है। तो इसका वाइल्ड टाइप क्या होगा, वेस्टिजियल विंग यानी कि शॉर्ट विंग्ड मेल। फाइन। नाउ द फीमेल, yes, and this is the male and their characteristics. So P plus is basically black, and this is the black, and the vestigial is the heterozygous condition. All right, and here is the gray and the this is black and long, and this is gray and vestigial. Fine. So what happens is, since most of the offsprings had a parental generation, that is the P generation phenotype, Morgan concluded that genes for the body color and wing size are genetically linked on the same chromosome. All right. So उन्होंने क्या देखा कि सारे मोस्टली जो ऑफस्प्रिंग्स थे उनका जो एक पेरेंटल फिनोटाइप है, ठीक है, तो जो हैड मतलब जो भी ऑफस्प्रिंग्स थे उनका जो फिनोटाइप मतलब जो कैरेक्टरिस्टिक्स थे वो मिलते थे पेरेंटल जीन से, ठीक है, या फिर पेरेंटल जनरेशन से। पेरेंटल जनरेशन क्या था, ये वाला, ठीक है। तो इससे मॉर्गन ने कंक्लूड किया कि जो जीन्स है, ठीक है, जो कलर और विंग साइज से रिलेटेड है, वो जेनेटिकली दे आर लिंक्ड ऑन द सेम क्रोमोसोम। तो यहाँ पे हमने देखिए उनके पास टोटल में 4 थे, ठीक है, जिनमें से एक तो सेक्स क्रोमोसोम हो गया, ठीक है, 3 हुआ ऑटोसोम्स। तो ये मॉर्गन कहना चाहते हैं कि वो किसी एक पेयर ऑफ मतलब सेम क्रोमोसोम्स में इन तीनों में से किसी एक में ये दोनों कैरेक्टर्स प्रेजेंट है, ठीक है। Wings of the Drosophila or the fruit fly. So now VG plus, that is this represents the normal wing, और normal को हम wild type भी कहते हैं, ठीक है। एंड VG को हम कहते हैं vestigial wing यानी कि recessive और short winged। All right, I hope till here it is clear. So now moving forward, what we see is that जो नॉर्मल विंग है वो वाली, जैसे मैंने कहा नॉर्मल को होता है वाइल्ड टाइप। यहाँ पे अगर म्यूटेशन हुआ, that means if in the normal type a mutation, all right, is caused, now what all are the possibilities that there can be the vestigial or the wing can be nicked, all right। By that I mean, see, it has formed basically a cut here, all right, that is termed as the nick, and now it can even be a notched wing, all right। Similarly, these are the possibility, or it can be like the wing can be represented as a strap here। So what happens, see, this is the entire process what we have discussed। So this is our parental generation। हमारा parental generation क्या था, होता हमारा वाइल्ड टाइप यानी कि नॉर्मल टाइप, ठीक है। नॉर्मल टाइप क्या था, ग्रे विथ नॉर्मल विंग्स और लॉन्ग विंग्स, ठीक है। एंड फिर आता है डबल म्यूटेंट, ठीक है। डबल म्यूटेंट यानी कि वाइल्ड टाइप से जो अलग है, ठीक है, जो हम मेंडल के टर्म में रिसेसिव कहते थे, उसमें होता है ब्लैक एंड वेस्टिजियल विंग्स। इन दोनों का अगर हमने टेस्ट क्रॉस किया तो जो F1 हाइब्रिड आता है वो इसी का जो फिनोटाइपिकली एकदम पेरेंट की तरह दिखता है, ठीक है। बट ये होता है हेट्रोजाइगस कंडीशंस, इट विल बी ग्रे एंड इट विल हैव नॉर्मल विंग्स, बट इट विल बी इन अ हेट्रोजाइगस कंडीशंस। ऑलराइट, yes, so, and here also we will get a, see, this is the heterozygous condition shown, for this also we will get Yes, and when the double mutant is crossed, we get the same parental generation as it was. Here there is a heterozygous condition, all right? But in this case, it is not. So now this is fused with the wild type, all right? तो जब इसे वाइल्ड टाइप के साथ फ्यूज करते हैं, मतलब ये हुआ हमारा F1 जनरेशन, एंड इसको हमने फ्यूज किया वाइल्ड टाइप के साथ जो हेट्रोजाइगस नहीं है, जिसके पास ब्लैक वेस्टिजियल विंग्स है, फिनोटिपिकली भी एंड जीनोटिपिकली भी वो हेट्रोजाइगस नहीं है, दैट इज होमोजाइगस, ऑलराइट। एंड इसमें इस हेट्रोजाइगस पेरेंटल जनरेशन को फ्यूज किया हमने एक वाइल्ड वाइल्ड टाइप के साथ, ठीक है। तो तब क्या हमें मिलता है, उसके जो भी ऑफस्प्रिंग्स आएंगे वो है सी। फर्स्ट विल बी सिमिलर टू द पेरेंटल टाइप, दैट इज it will have gray and normal wings, and the second one is similar to the other parent and it will have black and vestigial wings. Now the other two that are produced, this has gray and vestigial wings, ठीक है। ग्रे अभी किसका कैरेक्टर हुआ, ग्रे हुआ इसका कैरेक्टर, ठीक है, ये पेरेंट का कैरेक्टर, एंड वेस्टिजियल किसका कैरेक्टर था, ये वाला, फाइन। एंड अभी फोर्थ वाले में हमने क्या देखा कि इसके पास है ब्लैक, ठीक है, यानी कि इसका कैरेक्टर, और we have normal wings, that is the character of this. So basically we see a cross, all right, we see a fusion happening, fine. So this is the expected parental phenotype that we expected, and this there was a recombination, ठीक है, यानी कि यहाँ पे एक कॉम्बिनेशन कॉज हुआ जिसमें से दोनों के कैरेक्टर्स हमें एक में ही देखने मिले। इससे ये हम प्रूव करता है कि मेंडल ने भी एक इंडिपेंडेंट असॉर्टमेंट की थियोरी दी थी, बट उस टाइम पे ये वेरीफाई नहीं हो पाया था, ठीक है, क्योंकि पी प्लांट में या जब हम दूसरे प्लांट्स लेते थे तो काफी एक्सेप्शंस या फिर डिविएशन आते थे मेंडल के लॉस से। बट हियर टी मॉर्गन प्रूव्ड विद ड्रोसोफिला दैट विद द हेल्प ऑफ क्रोमोसोम्स, chromosome is the one that causes recombination, and what was expected is independent assortment, and this is the best example for independent assortment, and T Morgan proved that the theory of independent assortment of Mendel was right. All right, hi Sriraj, how are you? Hi Harman. All right. So how many pairs of sex chromosomes are present in a Drosophila? We see and whole Drosophila has 4 sets of chromosomes. Out of that, 3 are autosomes. All right. And 1 pair is the sex chromosome. All right. I hope this is clear. Now we will move to a very interesting concept of Cleotrope. All right. So, what is meant by pleiotropy? Pleiotropy means multiple effects of a single gene. यानी कि एक सिंगल जीन हमारे एक्सटर्नल मॉर्फोलॉजी या हमारे बॉडी में बहुत सारे इफेक्ट्स ला सकता है, ऑलराइट। सो व्हाट हैपेंस दैट वन स्पेसिफिक जीन कंट्रोल्स अ स्पेसिफिक फिनोटिपिक ट्रेट, जैसे कि हमने ड्रोसोफिला के केस में भी देखा कि एक स्पेसिफिक क्रोमोसोम उसका विंग कलर या जो विंग साइज है वो कंट्रोल कर रहा है, ठीक है। तो सिमिलरली इवन फॉर ह्यूमंस a specific gene will be assigned to control a specific trait. But in case of a pleiotropy, what will happen is that when there is pleiotropy, a single gene will have multiple traits on the body. Yeah. So a single gene will influence more than one phenotypic trait. Phenotypic trait मतलब जो हमारे बॉडी में जो चेंजेस हमें विजिबल हो, जेनेटिक ट्रेट नहीं, फिनोटिपिकल। जैसे कि सिकल सेल एनीमिया में हमें बहुत सारे कंडीशंस होते हैं जो हम आगे डिस्कस करेंगे, या फिनाइलक्यूटिनिया में सिर्फ एक जीन की वजह से हमें स्किन रैशेस भी आते हैं, हमें प्रोटीन रिलेटेड प्रोटीन लेवल्स बहुत हाई हो जाता है बॉडी में, ठीक है। देन देयर इज डिपोजिशन ऑफ फिनाइलएलेनिन, बहुत ज़्यादा फिनाइलएलेनिन डिपॉजिट हो जाता है। तो ये सब क्या है, मतलब सिर्फ एक जीन इतने सारे फैक्टर्स कॉज कर रहा है, जिसके हम आउटकम देख सकते हैं फिजिकली हमारे बॉडी में। इसे कहते हैं जीन एक्सप्रेशन। ऑलराइट, वो जीन अपने आप को इतने वेज में एक्सप्रेस कर सकता है। सो द सेम जीन इज एक्टिवेटेड इन several different tissues producing different phenotypic effects. ठीक है? So the example is cystic fibrosis, phenylketonuria, and sickle cell disease or the sickle cell anemia. All right. The first example is sickle cell anemia. All right. Please let me know if there is any doubts. Right. So basically, why do we say that the sickle cell anemia is pleiotropic? Because, see, why is sickle cell anemia caused in first place? It is basically caused because of the mutated, that is the mutated HBB gene. All right. So whenever the gene carries or the S, whenever there is this condition, all right. So what happens is they produce a numerous number of phenotypic traits, what is termed as consequences. All right. So this is the exact term. So what happens is one single gene, all right, one single gene when it is mutated, it produce a numerous consequences. Fine. And this consequences is seen throughout the body. All right. So what happens is that this mutated hemoglobin now will form polymers. All right. तो ये जो म्यूटेटेड हेमोग्लोबिन है ये नॉर्मल आरबीसी है और ये हमारा सिकल सेल आरबीसी है। तो बेसिकली उसका शेप चेंज हो जाता है। ठीक है। और ये जो म्यूटेटेड आरबीसी है हमारा, ये फॉर्म करता है क्लम्प्स। ठीक है। एंड जब ये क्लम्प्स फॉर्म करता है तो क्या होता है कि ये एक डी आरबीसी का काम क्या है, ऑक्सीजन ले जाना। बट ये क्लम्प्स इज कॉजिंग द डीऑक्सीजनेटेड आरबीसी। ठीक है। तो मतलब एकदम ही उसके काम से ऑपोजिट हो गया। एंड इससे हम मतलब बहुत सारे फिजिकल फैक्टर्स ये इफेक्ट करता है। राइट। If any doubts, please let me know. Hi Ayushi. सो जैसे कि मैंने कहा ये फॉर्म करता है क्लम्प्स और वो आरबीसी का काम होता है ऑक्सीजन बेसिकली कैरी करना एंड ये कॉज कर देता है डीऑक्सीजनेटेड ब्लड। ठीक है। एंड ये कॉन्सिक्वेंसेस में क्या आता है, सिकल सेल एनीमिया, पहली बात ब्लड में ऑक्सीजन नहीं है तो इट कॉजेज ऑक्सीजन फेलियर। ऑलराइट, इट कैन कॉज एक्सट्रीम फटीग, you will have urinary problems. So see, one single gene causing so many problems. All right, first of all it affected the RBCs, then it is causing you fatigue, tiredness, anxiety. All right, other factors, then we have urinary problems related to it. Then even the lungs don't get enough supply of oxygen. So there are so many traits related to it. All right. Now, coming to another important condition of phenylketonuria. So basically what happens at alanine, for फिनाइल हाइड्रोक्सिलेज क्या करता है, वो उसको कन्वर्ट करता है टायरोजीन में। सो दिस एंजाइम फिनाइल एलानिन हाइड्रोक्सिलेज इज वेरी इंपॉर्टेंट। ठीक है। फिनाइल कीटोनुरिया के कंडीशन में क्या होता है कि ये जो फिनाइल एलानिन हाइड्रोक्सिलेज है ये सेक्रेट नहीं होता। इससे क्या होता है कि नो फिनाइल एलानिन हाइड्रोक्सिलेज, इससे क्या होता है कि फिनाइल पाइरुवेट बन जाता है और टायरोजीन नहीं बन पाता। ठीक है। और कॉज करता है बिल्ड अप फिनाइल एलानिन का। तो बॉडी में फिनाइल एलानिन के ये बिल्डअप होने लगते हैं। ठीक है। So phenylketonuria mein basically what happens is that this condition is also known as PKU, and it is a very rare condition that is there, and in which the baby, when it is born, it is it does not have the ability to break down the amino acid that is phenylalanine. It is basically an autosomal recessive trait, all right, and basically it doesn't affect you, but there is a deposition of phenylalanine and it leads to multiple symptoms. It leads to fatigue, protein accumulation in body, and you have a multiple diet restrictions. And phenylketonuria is basically a genetic disorder, that is caused by the chromosome 12. This can be asked as a question. Please learn this entire line that phenylketonuria genetic disorder caused by the mutation in the gene PAH located on chromosome 12. Fine. PAH is phenylalanine hydroxylase. So this is how, as soon as the baby is born, if it suffers any of the symptoms, this is how the rapid test is carried out for the phenylketonuria, and this is the phenylketonuria test, and the proper conditions and the precautions are taken. Sickle cell anemia is an example of Cleotrope. Similarly, sickle cell anemia, phenylketonuria, also a very good example of Neotropy. So that is it for today. We have discussed how the chromosomal inheritance was T. Morgan's experiment in detail, and also how this theory was very much similar to that of the Mendel, and why this was more accepted. And we saw in detail what is pleiotropy, what does it mean, and it's associated to diseases, sickle cell anemia and phenylketonuria. So tonight we will be discussing a very interesting concept of pedigree analysis, and along with it, I will try to cover a few more topics from this same chapter. Mostly, we will cover all the autosomal dominant and the recessive genetic disorders, and the sex-linked disorder. Thank you so much for joining in. Uh, Please be online with me at 8:45 p.m. tonight. All right, we will be discussing a very important topic, and this chapter is one of the most important chapters which has a very high weightage for your NEET examinations. Thank you so much. Please do like the video, comment your views, and subscribe and hit the bell icon below. All right, so please, like, it is like a